Okay, so let's unpack this. If you wanted to map every single street, every alley and driveway in a massive metropolis, you'd probably launch a satellite, right?
Right, yeah. That's the standard approach.
And we've basically done that with the brain. It's called a connectome. But what if, to understand how that city actually thinks and allocates its resources, just mapping the streets wasn't enough?
Yeah, what if you had to look through the roofs?
Exactly. Looking through the roofs and mapping exactly where every single electrical generator was parked inside every single building, literally down to the inch.
Which is just a mind-boggling scale.
It really is. And that is the insane scale of connectomics we're deep diving into today. We're looking at this groundbreaking 2026 paper from Science by Sager and colleagues.
Yeah, and they moved past just mapping synapses. They mapped hundreds of thousands of individual mitochondria across entire neural circuits.
Hundreds of thousands. I mean, you probably know mitochondria as the powerhouse of the cell from high school biology.
Yeah.
But what if their exact shape, their size, and their location were actually the secret code dictating how your brain processes information?
Right, how it learns and forms memories. We are crossing a major threshold here, from mapping network wiring to mapping nanoscale cell biology.
And the data sets are just staggering.
They really are. We have the Drosophila hemibrain connectome, which contains about 25,000 neurons for the fruit fly.
Wow.
And then the Micron S dataset, which maps the mouse visual cortex. Historically, connectomes just showed us the chemical synapses, the junctions where neuron A talks to neuron B.
Just the connection points.
Exactly. But Sager's team used electron microscopy connectomics to actually look inside those neurons. They analyzed the whole organelle distribution across these massive networks.
And the immediate realization, the first major finding they hit us with, is that these organelles aren't just generic batteries floating around in the cytoplasm.
Not at all.
Their shape is actually a highly specific cellular fingerprint. It varies based on the cell type, whether it's in an axon or a dendrite, and even the specific neurotransmitter it's fueling.
Yeah, and to prove that at scale, they had to isolate a very specific testing ground first. They went straight to the lobula columnar neurons, or LC neurons, in the fly's visual system.
Right, because those are perfect for this, right?
Exactly. They share an overarching architecture for visual processing, like detecting moving objects, but they are subdivided into distinct types that handle highly specialized tasks.
So if form truly follows function at the nanoscale, these related but distinct neurons would show it.
That's the idea, but they didn't just measure length and width. They measured these mitochondria across a 21-dimensional morphometric space.
Okay, wait, 21 dimensions? That's hard to even picture. For you listening, to visualize 21 dimensions, try to imagine a microscopic 3D scanner mapping a single organelle.
Right, a highly detailed scan.
It's calculating total volume, its surface area, how perfectly spherical it is, its cross-sectional area, its aspect ratio, and its symmetry across literally every possible geometric axis.
Yeah, it generates this robust 21-point mathematical signature for a single mitochondrion.
But human brains, and certainly scattered graphs, cannot process 21 dimensions.
No, we definitely can't. So to analyze this, the researchers used an algorithm called UMP.
UMP, right.
Yeah, think of UM as kind of like casting a complex shadow. If you have a highly complex 3D object and you shine a light on it, the shadow it casts in a 2D wall preserves the relative relationships of its features, just flattened out.
Oh, that's a great analogy.
Thanks. So UM mathematically flattened this 21-dimensional data into a 2D visual map. And when they did that, distinct clusters actually emerged.
The mitochondria were grouping themselves.
Exactly, based on the exact type of neuron they belong to.
But wait, I'm looking at this next step, and my first instinct with any machine learning application is to suspect overfitting.
Oh, sure, that's a valid concern.
Because they brought in a random forest classifier, right, an AI, to predict the neuron type purely based on this shape data. How do we know the algorithm didn't just hallucinate a pattern and memorize the training data?
Well, what's fascinating here is how they avoided that classic machine learning trap. They used 5-fold cross-validation.
Okay, break that down for us.
They split their massive data set into 5 chunks. They trained their random forest on 4 of the chunks, deliberately hiding the 5th one.
Okay.
So the algorithm had to build its decision trees, basically voting on whether an organelle's asymmetry or wrapping structure belonged to, say, an LC10 or an LC16 neuron.
And then they tested on the hidden data.
Exactly. They applied those rules to the hidden chunk. And they cycled this process 5 times, and the results held up. It proved this isn't just statistical noise.
Let's talk about the F1 scores from that validation, because they're frankly wild. The algorithm achieved an F1 score of 0.60 for predicting specific LC neuron types.
Which is huge.
It is, because given the vast number of types, a completely random guess would only yield an F1 score of 0.05.
Right, a massive improvement over random chance.
And when they expanded the AI's task across all the different brain neuropills, it scored 0.38 compared to a random baseline of 0.04.
Yeah, and we also see massive structural divergence based on the neurotransmitters themselves. For instance, GABAergic cells, which are part of the inhibitory network, contain significantly smaller mitochondria compared to dopaminergic cells.
And the algorithm just picked right up on those physical constraints.
It did.
And this isn't just a fly thing either, is it? Because they ran the exact same classifier on the mouse cortex dataset.
Yeah, the Micronis dataset.
They asked the AI to separate excitatory pyramidal cells from inhibitory interneurons using literally nothing but mitochondrial shape.
Yeah.
And it hit an F1 score of 0.99.
Yeah, 0.99, near absolute perfection across species.
That's mind-blowing.
It really implies a deep evolutionary conservation. The energetic demands of firing an excitatory signal versus maintaining an inhibitory baseline, it literally physically molds the organelle.
It's so distinct that you could take a tiny 50-micrometer fragment of a severed dendrite, feed its mitochondrial shape into the algorithm, and identify the exact identity of the host neuron.
It's a true cellular fingerprint.
Okay, so before we transition into the next piece of this puzzle, let's consolidate what we've unpacked in this first segment. Let's do a quick 3-point recap to lock this in.
Sounds good.
Point 1, connectomes have evolved from mapping external synapses to detailing internal organelles.
Yep.
Point 2, mitochondria possess 21-dimensional shape profiles, and we use dimensionality reduction like UMP to visualize these signatures.
Exactly.
And point 3, machine learning proves that mitochondrial morphology acts as a strict cellular fingerprint, accurately predicting cell types and neurotransmitters across entirely different species.
Spot.
So think about your own highly specialized daily routines for a second. If an algorithm could look purely at the physical tools you use, the engine of your workflow, how easily could it predict your exact profession?
It's a great question of specialized mechanics, but, having a highly specialized engine is completely useless if it's sitting in the wrong part of the factory.
Exactly. Which brings us to the next massive hurdle in this data, location. We know the shakes are specialized, but how do these powerhouses know exactly where to park on the winding highways of our neural wiring?
Right. How do they find their workstation? So the researchers approached this by building a multivariate logistic model.
A probability engine, basically.
Yeah, exactly. They took the actual observed locations of the mitochondria and compared them mathematically to completely randomized shuffled locations within the same neural branches.
Looking for the hidden zoning laws of the cell.
Precisely. And to find the precision of those zoning laws, they ran a jittering experiment.
Oh, I love this part. Jittering literally adds artificial noise to the coordinates, right?
Yes. In the computer model, they digitally bumped the mitochondria off their actual locations by just a few micrometers.
It's just a tiny nudge.
They shifted them anywhere from 0 to 6 micrometers, and when they fed that jitter data back into the logistic model, the predictive accuracy completely fell apart if the organelles were moved just 2 to 3 micrometers from their original spots.
Wow.
The probability engine failed because the underlying biological logic was broken.
Here's where it gets really interesting. It's the equivalent of parallel parking a massive semi-truck perfectly on a microscopic street. The precision is just astonishing. But if 2 to 3 micrometers is their operational window, what are the specific rules drawing them in?
Well, the logistic model revealed several heavy attractors. They are heavily biased toward presynaptic sites, which are the specific active zones where chemical signals are released.
Or signals are sent out.
Right. They also preferentially park in larger daughter branches in the dendritic arbor, and they cluster densely at the base of axons.
Probably because initiating an action potential carries a massive metabolic cost, right?
Exactly. It needs power right there.
But there's also a physical pushback, isn't there?
Yeah.
A repulsive force.
Yeah, and we see a fascinating cross-species divergence here. In the fly axons, mitochondria actively repel each other through steric hindrance.
Steric hindrance, let's define that.
It's a concept from physical chemistry. It essentially means molecular elbow room. You cannot occupy the space another physical mass is already occupying.
Makes sense.
So in the narrow fly axons, they space themselves out perfectly to avoid clashing. However, in the wider mouse cortical neurons, the mitochondria actually show a tendency to cluster together at those high-demand sites.
But wait, I need to pause on the mechanics of this parallel parking for a second, because
Yeah.
I always pictured mitochondria as highly mobile. We've all seen those fluorescent imaging videos of them zooming up and down the microtubule tracks of an axon.
Yeah, the classic textbook videos.
Right. If they're constantly in transit, always moving, how can they have a fixed 2 to 3 micrometer precision? What throws the emergency brake?
This is a critical distinction between the mobile fraction and the docked fraction.
While a certain percentage of mitochondria are indeed in transit, acting as a roving supply line, a massive portion are predictably immobilized. Specialized docking proteins effectively anchor them to the microtubule tracks, exactly where they are needed.
So they're locked in.
Yeah. Through simulations, the researchers deduced that the high accuracy of Sager's model means the EM scan captured these mitochondria in their docked, immobilized state when the brain was preserved.
So the mobile ones are just statistical noise.
Exactly. The docked ones reveal the true underlying rules of energy demand.
It's like a freeze frame of a bustling city. The model proves that the food trucks aren't just driving around randomly. The vast majority are parked exactly outside the busiest office buildings with their parking brakes firmly engaged.
I like that analogy.
Yeah.
And those parking spots are dictated by the cellular economy.
Let's do our second quick 5-minute recap here to keep our framework solid.
Go for it.
Point 1. Mitochondrial placement is not randomly scattered. It follows strict positioning rules via a multivariate logistic model.
Point 2. Their spatial precision is locked into a tiny 2 to 3 micrometer window, which we know from the jittering experiment.
Correct.
And point 3, they are drawn to presynapses and thick branches, but repel each other in flies while clustering in mice, and they rely on docking mechanisms to stay put.
Perfect summary.
So a question for you listening. If your energy was placed as deliberately as these mitochondria, what high-demand synapses in your life would you park your resources next to?
It definitely forces you to look at resource allocation. And if we connect this to the bigger picture, that brings us to the most profound question addressed in the paper.
Why does the brain arrange its power grid this way?
Exactly. We have the physical maps, we have the zoning laws, but how does this tie into actual thinking, learning, and circuits?
To crack that, they had to correlate this static map with spontaneous brain activity.
Right. They integrated data from previous in vivo studies that used fluorescent indicators to measure calcium, ATP, and pyruvate.
And they found a direct, measurable correlation. Brain regions with higher fractional standard deviations in activity, meaning regions that fire more frequently and intensely, had a correspondingly higher mitochondrial coverage.
And just to clarify, mitochondrial coverage is the exact fraction of presynapses that are physically in contact with a mitochondrion.
So the more a circuit fires, the more its synapses are physically backed up by a dedicated power plant.
Yes. And we also see this very heavily by the type of neurotransmitter system. Serotonergic neurons, which modulate widespread brain states, have the absolute highest mitochondrial coverage at their synapses.
Oh, wow.
While dopaminergic and octopaminergic neurons have the lowest.
But the absolute mind-blowing finding comes when we zoom in on the fly's mushroom body. Let's talk about the mushroom body.
The learning center.
Right. This is the anatomical center for associative learning. It's how a fly learns to associate a specific odor with a reward or a shock. And the architecture here relies on Kenyan cells.
Kenyan cells, abbreviated KCs. And the mushroom body is heavily compartmentalized. It is divided into 15 distinct functional compartments.
15 zones.
Yeah. A single Kenyan cell axon acts almost like a main power line traveling through several of these compartments sequentially.
And the data shows something that completely upends how we think about cellular autonomy.
Yeah.
Within that single Kenyan cell axon, the likelihood of a mitochondrion parking at a specific synapse depends entirely on who the postsynaptic partner is on the receiving end.
It is entirely dictated by the local environment. The Kenyan cells synapse onto mushroom body output neurons, or MBONs.
MBONs.
Yes. These MBONs act as the steering wheel, dictating whether the fly approaches or avoids a stimulus. Sager's team discovered that mitochondria are dynamically enriched at the synapses connecting to very specific MBONs, depending on the compartment.
It's like having a smart home where the electrical grid automatically routes all the power to the specific smart plugs connected to your heaviest appliances, but only when you're trying to learn a new recipe.
That's a really good way to visualize it. This implies retrograde signaling. The receiving neuron is somehow signaling backward across the synaptic cleft, telling the transmitting neuron, I am a high-priority connection, drop an anchor, and park a mitochondrion right here.
That is wild. And they didn't just trust the dead EM tissue for this either, did they?
No, they didn't. To ensure this wasn't an artifact, they validated it in vivo. They used 2-photon microscopy on living flies, tagging the mitochondria with a fluorescent green marker and the cell membranes with a red marker.
So they watched the power grid in real time.
They did, and they proved that these density differences dynamically exist in real life, in living, breathing, learning networks.
There is a beautiful control case in the data that proves this environmental rule, too, the APL neuron exception.
Ah, yes, the anterior paired lateral neuron. It is a massive, giant inhibitory neuron that connects everywhere, stretching across all 15 compartments of the mushroom body.
So if mitochondrial placement was dictated strictly by the cell's internal genetics, the APL neuron would have a uniform distribution everywhere.
Exactly. But instead, its mitochondrial coverage perfectly matched the local compartmental rules of the Kenyan cells it was sitting next to. The neighborhood dictates the plumbing, regardless of the house.
Which raises the ultimate pushback question, the chicken or egg scenario. So what does this all mean? Does the brain build this highly specific power grid first during development, and that dictates our capacity to learn? Or does the act of learning pull the power grid to the synapse?
The evidence strongly points to the latter. If we connect this to the bigger picture, mitochondria are the foundational requirement for synaptic plasticity.
The ability to change.
Right. Plasticity, the ability of synapses to physically grow, strengthen, or weaken over time, is the biological basis of memory. It requires massive amounts of localized ATP to synthesize new proteins and expand the synaptic membrane.
So this connectome we are looking at is not just a static blueprint.
No, it is essentially a structural fossil record of the brain's past learning and spontaneous activity.
The energy follows the demand.
Yes. Spontaneous activity and learning create metabolic demand, which triggers the docking mechanisms, halting the mobile mitochondria. By docking there, they provide the energy required to lock in those specific memories.
Let's do our final 5-minute bullet recap to bring this all home.
Let's do it.
Point 1, mitochondrial density predicts regional brain activity with a heavy reliance on the neurotransmitter type.
Right.
Point 2, in learning centers like the mushroom body, synapses are assigned mitochondria based entirely on exactly which output neuron they are talking to, driven by the local environment.
Mm-hmm.
And point 3, this distribution is a physical mechanism for associative learning and memory.
Beautifully summarized.
So, to you listening, what stands out to you? Does it change how you view learning, knowing that your cells are literally moving power plants around to support a new memory?
It completely bridges the gap between the abstract concept of a memory and the heavy physical resource cost required to maintain it.
We started today looking at a static, highly detailed map of neural wiring. We zoomed in to find 21-dimensional power plants acting as cellular fingerprints. We discovered their strict 3-micrometer parallel parking rules. And we ultimately realized that this entire microscopic grid is actively shifting to power our learning and memories.
And I want to leave everyone with a final, lingering concept to mull over. If mitochondrial docking is deeply tied to synaptic plasticity and memory formation, then the act of you listening to this very deep dive right now is physically drawing mitochondria down the microtubule tracks of your neurons.
Oh, wow.
Your memory of this deep dive isn't just a software update. It is currently existing as a literal structural shift in your brain's power grid.
You are physically rewiring your power grid just by analyzing this data with us. Thank you for joining us on this deep dive, and keep exploring the incredible nanoscale architecture of your own mind.